you can have a good gene, but if that good gene doesn't produce a functioning gene product, it doesn't matter. So that gene product either needs to be there, and it needs to be there and working. So now incomplete dominance. A heterozygote that is heterozygote for an incompletely dominant trait will be what? Incomplete. So neither trait can kind of overpower the other one. So what does that mean? I'm sorry? Oh yeah, they're both, so the heterozygote, so it has one trait for one trait for the other. But it's incompletely dominant, so neither trait can beat the other one. So what does that mean the individual is going to look like? Good, it's going to be a mix. So it's going to be a blend or an intermediate. So it's a blend or intermediate of those two traits where neither trait can kind of beat or overpower the other one. And then pleiotropy, so what in pleiotropy causes what, if I could make that any more vague? Any ideas? So let's say this is a mutation. So one mutation causes what? Any ideas? Good, I think I heard it. So it's going to be multiple phenotypic effects. So if you mutate something in your genome that codes for a really important protein, you're probably going to see multiple effects of that mutation as opposed to a small protein that's not used a whole lot. Okay, so the other part of incomplete dominance is codominance. So I like to think of this as the two genes are working together, so they're functioning, they're like co-managing kind of. So the phenotypic effects of two allelic genes are what? How are they expressed in codominance? Good, at the same time or simultaneously? Okay, and epistasis. This is another concept. So the effect of one gene does what to another gene? Good, it masks or it's going to modify. Mask or modify another gene. Okay, so penetrance and expressivity. They're kind of the same, but they're kind of not. Yeah. Good question. So it all has to do with what you're seeing in that offspring. So if you know a parent looks, I guess the most popular way of looking at this is the flower snapdragons. So there's red and white purely. So you're going to have a red and a white parent, and then you have offspring, and those offspring can be one of three things. So you only had two phenotypes in the parent, but the offspring can be red, white, or pink. So once you see a new phenotype that's kind of like an in-between of what the parents look like, that's incomplete because neither one of those traits could beat the other one. When codominance, you're going to see both of those traits kind of working together to both be expressed in that individual. So you would see both red and white, not the intermediate of pink. Does that kind of explain that? I kind of get into that a little bit later. Yeah, definitely. Please let me know if you have questions. Penetrance and expressivity. What's the difference? What's the same? How do you look at one versus the other? Let me get started. Okay, so I think of penetrance as all or nothing. This trait is either going to penetrate the entire population and every individual, or it's going to be missing in some or all of the individuals. So if it's a color, every single individual is going to have that color, or they're not. There is no other option. They do or they don't. When expressivity is a little bit more forgiving, in that you can have a range of phenotypes. So instead of having all or nothing, you might have one color, you might not have the color, or you might have somewhere in between. I'm trying to think. I know that the gene that they discovered on the genome for human breast cancer is variable penetrance. So that means everybody has the gene, or all females have the gene, but sometimes it hits you and sometimes it doesn't, and they don't know why. So that's an example of incomplete penetrance. So it's everybody has that gene, but not everybody gets cancer. And expressivity is kind of similar to incomplete dominance, but in incomplete dominance you only have no. Okay, maybe I hit it. No, that's okay. It wasn't me. Good. Okay. Any questions on penetrance, expressivity? No? Okay. So now we go into blood typing. So ABO blood types. So type A and B, what kind of, what have we talked about that explains type A and B? Yeah, that's what I was getting to, codominant. So when A and B are together, they work together to create an individual that is type A and B. And what type of antibodies does an individual with type A blood have? Good, they have anti-B antibodies. So that means if a type A person gets type B blood, they will attack the type B blood. Where type B has what kind of antibodies? Good, anti-A, you can't give a person with type B blood A because their body will attack it. And what about type O? Good, they have both. You can't give O, A, or B because their body will attack both. Okay, so this is the genotype and phenotype of an individual with the Bombay phenotype if the parents were both homozygotes, one for A, one for B. So what's the genotypic offspring? The genotype of the offspring of this cross. Good, AB. You would have AB blood, one A from mom or dad, the other from the other parent. And if they have the Bombay phenotype, what are they going to express as? Their phenotype is going to be O. Because they don't have the necessary, I think it's antigens, I'm not sure, to make the antibodies up here. So they can't make these antibodies, which means that they don't have them, or they can't fight them, I guess. So they have type O, which they have both of them, so you can't give them type A, B, or AB blood because they don't have what's necessary to make that blood okay in their body. So anybody with a Bombay phenotype, regardless of what they are phenotypically they present as O. Okay, and now true, false. A sex-influenced character only occurs in one sex. Remember, we have sex influence, and then the other one we have is sex limited. That's our other option. So is sex influenced or sex limited only in one of them, male or female? Sex limited. So this is false. A sex influence trait can occur in both sexes. It's just different in how it expresses. In a sex limited trait, it would only occur in either males or females. Another true, false. A conditional allele will express a normal phenotype under permissive conditions and not express it under restrictive conditions. Does that make sense to you? Yeah? Okay, so this is true. If an allele has a condition that it needs to express, that condition will work under permissive, and it will keep it from being expressed under restrictive. Even if you don't know that concept, those words should kind of clue you in that restricting will restrict the allele, permissive will allow the allele to express itself. Okay, and what is the effect of methylation on DNA? This is the concept of genetic imprinting. Good, silence. So we're going to silence it, or we're going to decrease transcription. So methylation prevents enzymes from attaching to DNA and coding it, transcribing it, all that stuff. So if we methylate DNA, we're not going to have as much transcription. Or we're going to silence that DNA. It cannot become any kind of functional protein. So this is an example of an incompletely dominant question. So again, I'm talking here about snapdragons, which is a type of flower. And there are 3 possible colors of snapdragons, red, white, or pink. And it's asking for the genotypes of both the parents and the offspring in these 4 crosses. So if we cross a red flower with a red flower, what are we going to get? Good, everybody is going to be red. And whatever you choose to be red in, I guess, representation in Punnett square is just going to always be red. That's everything. What about white and white? They're always going to be white. There is no variation. What about white and red? This is incompletely dominant, remember. So if these genes interact, neither of them can win. They're all going to be pink. And if we have pink and pink, now we see the difference. So we're crossing heterozygotes here in this case. So if you think back to our cross of a heterozygote, we're going to have, go ahead. Good, you're going to see all three. So you're going to have 1/4 of them, or 1 out of 4 are going to be red. 1/2 are going to be pink. And the remaining 1/4 are going to be white. And this is a note that happened during exams. Remember, if he gives you a lethal allele or a lethal gene, don't use the individual that has the lethal allele or gene in a mating experiment, because that's not okay. So they are dead, you can't use them, there are no offspring if the lethal allele is present. So that's just a side note. Any questions on this? I know I didn't do the punnets, but it's kind of straightforward. Okay, so now pedigrees. So I gave you a ton of pedigrees because that was the #1 request that I had for more practice on. So I don't expect you guys to have to go through and work each of them. If you worked on it beforehand, that's awesome, and you can shout it out, and I will ask. But if not, I'll give you what I got for it. But remember, there are sometimes multiple explanations for pedigrees. So if you do something else and you find out it works, it might also work. This is just what I discovered when I did it. So does anybody practice these beforehand or do anything beforehand, just so I know? No? Okay. When I did this pedigree, I found this to be an autosomal dominant pedigree. And if you want me to go through any of them, I can. I'll pick and choose some to go through. So this I found to be an X-linked recessive trait, or a pedigree. This is an autosomal dominant pedigree again. And this is an autosomal dominant pedigree again. So of these 4, is there one that you want me to go over or go through so you kind of know the process of working any of them? I'll pick one of the 4, but if anybody just wants me to do one. No? I'll do one of the dominant, autosomal dominant, because that's sometimes hard to figure out. So let's just do the first one here. Okay, so it's good to know the different characteristics of each of these pedigrees. So in an autosomal dominant, you should see it in every generation. And your numbers of females and males should be about the same. If they skip generations or it's obvious that the males outnumber the females or vice versa, it's probably X-linked. Or if it's not in every generation, it's probably recessive. So every generation, males equals the females, and then you just kind of pick one or the other to go with. So even with this alone, you might be able to guess on the exam and get it right. So if you just start working through things, you can see each of these individuals should have this big trait, so a big A, the dominant trait. You can see one of the individual parents is not affected, which means they have a recessive trait, and they are heterozygote. Same thing for this individual. They have one parent affected, one parent not, which means they have one of the alleles that gives them the trait and the other one that doesn't. And that follows through for every single one of these individuals. Every affected individual has a heterozygote parent. And that is why this works. Maybe there's something else that works as well, but you can't rule autosomal dominant out in this situation. Yeah? It's kind of assumed that homozygous dominant for autosomal not X-linked is lethal typically. So you can make the assumption that you're never going to see anybody that is autosomal homozygous dominant. But that's definitely not true for X's. So don't assume that for X's, but you can sometimes assume that this is the case. Yeah. And here's even more. Yeah, definitely. Okay, so this one is autosomal dominant according to me. So let's see, in this case, it follows the same kind of pattern where each affected individual comes from a heterozygote parent. The trait is present in every single generation, and the numbers of males equals the number of females about the same. So if you just pick one individual and follow it through, they have the dominant, oh, you can't see. They have the dominant trait from their dad, and then they have the recessive trait from their mom in this case. Same thing for this individual. They got the dominant trait from their mom, the recessive from their dad. Same for this individual. Exact same thing for this person. And then if you go to the other side, it's the same. One from mom, one from dad, one from dad, one from mom. And this is somebody you could say you don't know if they are homozygous dominant or heterozygous, but that's when you would make the assumption that most homozygous autosomal dominant traits are lethal. So you would maybe say your best guess is that they have a little a, but you can't say for sure. So I think this works as a potential pedigree example for autosomal dominant, but again, there might be something else that works as well. And if you see that there's kind of an equal number of, males passing it to their daughters, females passing it to their sons and daughters, and that all works, that indicates autosomal as well. Anybody can give it to either son or daughter. Okay. So here's another just page of pedigrees, and I would encourage you to try to work through some of them. This is a pedigree that I worked out to be autosomal dominant. This is another autosomal dominant. This is X-linked recessive, which again you can see because nobody's affected here. We have an affected generation, not affected, same thing here. It's skipping generations. Not all of the time. In this pedigree, it just happens to only affect males, but X-linked recessive is much more likely to affect males because males only need one bad X or one recessive X to have that trait, where females need two. So it's much more statistically probable that a male is going to have an X-linked recessive disorder than a female because they only need one shot. Okay, and this is going to be an X-linked dominant. This is another autosomal dominant. This is going to be Y-linked. So this is a case where it is all and only males. So here you should always follow the lineage. So you do see some unaffected males here, but that's because they're not part of the actual familial tree. They married into the tree and then had children. So every single familial person that is a male has the trait. Again, same thing here. These individuals kind of married in and brought in new genes, which is why they're not affected. And then down here, I have X dominant. Is there any of them you want me to go over? I only have, I think, one X-linked recessive in here, but I kind of talked about that. And then a few X dominants, and then lots of autosomal dominants, because those are the hardest ones to determine. Yeah, definitely. Okay, so we see here it's in every generation, which should indicate that it is a dominant trait. And I kind of lost some of the pedigree there. So it's in every generation. And then we see here that we have a little bit more females than males, but not really enough to indicate that it would be X or Y. So you might start with autosomal, and then maybe it doesn't work out and figure out it is X-linked. So let's see. All of our males here should have one X with the dominant trait. So that should be each of these. And let's see. We know that this unaffected father gave his Y to his son, which means that if this son is to be affected, it must have come from his mother, which does make sense because she is affected. And the same thing here. This daughter, let's see, what is her genotype? We know she got an affected from mom. What did she get from dad? Unaffected, good. So she would be a heterozygote, so you would know her full, complete genotype. And let's see, this mom here should be the same because the father here is unaffected. So she should also be a heterozygote. We can keep going back up. Oh, I blocked off some of this. I'm assuming this is an unaffected female. Okay, so you see here, and it does make sense that this male is affected and his son is not affected because he only gives his son his Y, and this mother is not affected, which means her son should not have the trait. And each of these females is going to have the big A, or the affected dominant trait, from this affected father. But they would have a normal or unaffected X from their mother in this case. So this does work, this does make sense, but again, there might be something else that works as well. It's not always one or the other. Anything, any other pedigrees on this you want me to go over? No? Okay. And then I did a really brief linkage section because you're not likely to see a whole lot of linkage because you don't have calculators on your exam. So here's an example of a chromosome, and is this chromosome and its genes in a cis or a trans conformation? Good, it's in trans, and the synonym for that would be coupling or repulsion. Good, repulsion. So trans is the same thing as repulsion, coupling is the same thing as cis. So in this case, because we have our wild type here and a mutant here, and a wild type here, and a mutant here, we have kind of a flip-flop. So the two X's that want to be by each other aren't, so they're on opposite chromosomes, and the two mutants that kind of want to be by each other, it's kind of how I think of it, aren't next to each other. So when you have two unlike genes next to each other, they're repulsed by each other, so that's why they're in repulsion, where if you have two genes, like if you were to have the plus and the plus up here, they're like a couple, they belong together, so that would be a coupling example. Here is a true-false. As the distance between gene increases, their probability of crossing over decreases. True or false? Good, false. If we have two chromosomes, or examples here, and on this chromosome we have this gene and this gene, and we have then on this chromosome this gene and this gene, the likelihood of crossing over occurring is way more likely on this one, because the cuts could be made anywhere in between those two genes to separate them. Compared to this chromosome and these two genes, there's only a few places in there where the gene or the chromosome can be cut to separate those genes. So as the distance increases, it's more likely they're going to cross over and not be with each other on the same chromosome. Okay, so here is another type of problem. So this is asking you to use a branch diagram and to calculate the phenotypic ratios of the progenies from I just put one of these crosses on here. Okay, so in this case we have our H factors and then our blood type. So the blood type of this individual is A, blood type of this individual is AB. Now this individual, what's their phenotype? If they had two little H's. Good, their phenotype is O. So although their genotype does say that they're A, if they have two little H's, which means they don't produce the H factor, and they can't produce those antibodies, that means they have type O blood. So again, let's start off by using more simply the H factors, because there's no codominance involved in that. So in this kind of cross, all we're crossing is this. So what are we likely to see in our potential offspring? What kind of ratio or percentage, fraction? Good, 1-to-1, half-to-half, 50-50. So there is a half of a chance that we are going to have our two little H's, and then also half of a chance that we are going to have a heterozygote. And then from there, we just use a branch diagram to use the product rule to figure out what our potential offsprings are. So again, we're working with A and AB genotypes. So what about individual 1 here? What can they give to their offspring or their gametes? Good, they can only give A. So every single offspring is at least going to have one A allele from that parent. And then we go to the other parent. They can give either an A or a B. So in this case, they might give an A, but there is an equal chance they would give a B. And same thing down here. Could give their A, could also give their B. And in this case, what are the chances of giving an A or a B? They equal, different? Okay. So these should be your crossovers, so you just add those numbers, and you would get 25 math units. Okay, so it gives you a lot of information, but all it wants you to do is construct a linkage map, and you don't even really need to have a whole lot of information to do that. So what's the first thing you look at when you're making a linkage map? do the same thing you look for when you're finding the configuration of alleles and all that stuff. Biggest number, right? You got to find the biggest number. So here's our biggest number, because remember this also equals map distance or map units. So our biggest number are going to be the two genes that are the farthest apart. So they're 44 map units apart. So you make your chromosome and you put DP, which it tells us is for dumpy wing, and then CL, which is for clot eye, far apart. And then you're left with only one other gene, AP, for apterous wing. Which gene is the AP allele closest to? Good, CL. Smallest number, fewest map distances, AP would be closest to CL. So this distance is 4. And let's see, DP and AP are going to be 39. And then DP, CL would be 44. And then I didn't give you any questions for population genetics because that's using a lot of decimals and fractions and stuff, so I don't think he's going to make you do anything. But it is just good to have in the back of your mind the equations you need to know and what those letters stand for in case it gives you more of a conceptual question. So this has to do with, let's see, alleles. This has to do with genotypes. So every allele should always in the population come to 100% or 1, because every allele should be represented, whether that means there's 2, 5, 10, however many alleles there are. And then each of these, P squared should be your homozygous dominant, 2PQ heterozygote, Q squared homozygous recessive. Everybody should be one of those things. So when you add up all of those individuals, you should get 100% of that population. And again, I put here there's no calculator, so you're probably not going to see 3-point test crosses or Hardy-Weinberg equation problems. And then once we get into bacteria, that's when he started talking about experiments. And so for every experiment that he covered, know the design of the experiment as well as what the results mean and why they're important. So here's a true-false. And essentially what it's asking, is it true that phototrophs require nutrients added to the medium and auxotrophs can survive on minimal? Is that true? True, false. Okay, well, it's false. I'll let you know. Prototrophs are the ones that can survive on minimal medium. Auxotrophs are the ones that need something added to survive. Okay, and what was the setup of the Davis experiment? There's one kind of important design piece they used that was very important in demonstrating what they demonstrated. So any ideas? No? Well, they used a U-tube, not like the website, but an actual tube that looks like a U, that looks like this. And they demonstrated a certain requirement of recombination in bacteria. And do you know what that is? No? Good, exactly. There is a physical contact requirement. So if bacteria can't touch each other, they cannot recombine their plasmids. So by using this U-tube, they could keep them separated at certain times and then also let them interact in other times. And they found out that it is required that the bacteria can physically interact in order to recombine their plasmids. Okay, so there are 5 steps to what we were just talking about, recombination, and it's called conjugation, which is essentially just bacterial sex. And that connection they make is called the sex pilus. That's how they connect with one another. So in this example, we'll say the sex pilus is going to form between an F plus and an F minus cell. And you can remember that the plus and minuses have to do with certain types of plasmids. The plus cells have the plasmid, the minus cells don't have the plasmid, but they want it. So they're going to conjugate in order to give the other bacteria the plasmid that gives it the plus notation. And then the second step is the DNA strand of the F factor is nicked at the, it's called the ORI T, and it's just the origin of replication. Then that DNA strand is separated and it moves through that sex pilus to the F minus cell. So it literally gives it to the other cell. And then the next two are just statements where that complementary strand for the F factor is synthesized in the recipient. And then the donor cell, the F plus cell, also can resynthesize that single strand that it gave away. And then once it's over, those circles close, and now both cells are F plus. Or they have the factor in both cells now. Okay, and in bacteria, what does the R determinant do? I don't know. Antibiotics. Resistance to antibiotics is what the R determinants in bacteria are for, and that's why it's super important for you to always take all of the antibiotics you are prescribed, because if you stop short and save some for later in case it comes back on you, you're essentially selecting for the bacteria that have this R determinant, and now that bacteria can donate to every single other bacteria through conjugation the antibiotic resistance, and that's why some people become resistant to penicillin and it no longer works on them because they didn't take all their drugs, and now all the bacteria in their body are resistant to penicillin. Okay, and another part of the chromosome is the RTF segment, and that is called the resistance transfer factor. And it's just the component that's necessary for bacterial replication. Okay, so here's some examples of conjugation and what the results mean. I'm not going to get into exactly what F and HFR are, but I encourage you to do that on your own. So if this kind of interaction takes place, which is the example I used in the conjugation steps, that means we're going to end up with two cells that are F plus. If we have an HFR and an F minus interaction, there's going to be no change. One cell is still going to be HFR, the other cell is still going to be F minus. And if we have this kind of interaction, an F minus and an F apostrophe, you could say, we have two F apostrophe cells, or two F prime cells. And where can transforming DNA come from? So where does this interaction come from? Well, it can come from other cells or other types of bacteria. So it can come from another cell. This is also how viruses can infect bacteria and humans. And it also can just come from the environment. So there are sometimes a cell can die, and then its DNA can leave the cell, go into the matrix, and then other bacteria can pick up that DNA and incorporate it into their own genome, so that DNA comes from the environment they are living in. Okay, and here are a bunch of experiments talked about in the DNA portion. We only have about 30 minutes left, so I'm going to go ahead and ask if anybody wants me to skip ahead to any kind of specific chapter they want me to go over. I'm not sure I'm going to have time to get through everything. Other than that, I can just keep going in order, or I can skip to the end. Anything you guys want specifically? Yeah. Gene regulation? Is that? Yeah, sure. Definitely. Okay, so we'll skip ahead to gene regulation, which I think is the second to last chapter besides biotechnology, which is kind of just a for your information chapter. I can't do that, no. But you can review this all again online, and all of this came from the lecture of section 2. So if you're in section 2, everything I used came from the notes. Okay, so first, the thing we talk about in gene regulation, or the main thing we talk about, are operons. So the first operon is the lac operon. That's the most important one. If you want to know and have to just study one operon, make it the lac operon. Okay, so the LAC-Z gene, let's see, let me skip ahead over here too. Codes for beta-galactosidase. LAC-Y codes for permease. And LAC-A codes for transacetylase. And know what each of these do. So what beta-galactosidase does, permease and transacetylase, what they do, although this one is kind of unknown. We just know that it's necessary for the operon to function, but we don't know really what it does. Oh, and I skipped something. Is the lac operon typically on or off in a bacterial cell? Good. It's usually in the off position, and it's turned on under certain conditions. So Z, Y, and A are structural genes. That's super important to know. And then there also are control regions, not necessarily genes. So the LAC-I is the repressor. So that actually is a gene that codes for the repressor. The LAC-P is going to be P for promoter. And you know from previous chapters that that is where RNA polymerase binds. And then the LAC-O is going to be the repressor site. So it's where that repressor is going to bind when it wants to repress the operon. What's the inducer of the lac operon? Good, allolactose. So that's the inducer, which means that it inactivates the LAC-I coded for repressor. So typically that repressor is in place, but the inducer can come in, make that inactive, and turn the operon on. So here's an example, or kind of the flow of information on how this operon works. So we have low glucose or no glucose in the cell, which inactivates adenylate cyclase activity, and that activity is going to hydrolyze ATP, and that is essentially for energy. That's all that's for. Whenever you hydrolyze ATP, you're just putting energy into a system. So you're hydrolyzing that ATP, and you're creating CAMP and 2 phosphate bonds. Again, those phosphate bonds are simply for energy. And this interaction, CAP plus those high energy phosphate bonds, is going to activate CAP, C-A-P, and that will be the facilitator of RNA polymerase binding to the promoter or LAC-P region of the operon. So this is everything that has to happen in order to get the operon functional. Obviously, you also need lactose, which is why it's called the lac operon. And you need that lactose to be able to be broken down by LAC-Z and all of these things. But these are kind of the behind-the-scenes aspects of the lac operon. Okay, and then we have the tryptophan operon. Okay, the tryptophan operon, when it is under the condition where there is no tryptophan in the cell, that means the repressor is going to be in an inactive position, and the operon is going to be on. So when there's no tryptophan, that means the operon is going to be working and on. If there is tryptophan present, that means that we don't need to make tryptophan. So it's actually tryptophan itself that's going to bind to the repressor to activate the repressor and then turn the operon off. And tryptophan is an amino acid. It's not necessary to know that, but sometimes it's nice to know what the operons are talking about. So this is an amino acid synthesizing operon. You always need tryptophan. Everything needs all 20 essential amino acids. So this is just a backup system to make sure if you're not consuming tryptophan, you can make it when you need it. And the ARA or ARA operon is almost identical to the lac operon. It just uses arabinose sugar instead of lactose. And I wrote on the back of this, which is why it kind of looks weird. So there's, if the situation is there's no glucose in the cell, but there is arabinose, then you need, there's 2 steps to inducing the ARA operon. So arabinose is a sugar, and that sugar is going to bind to ARA-C to produce something called I, and that's just a product of the gene. So arabinose sugar is actually going to bind to this region of the operon, ARA-C, and produce and cause I to be produced. And then you're going to have CAP, just like you do in the lac operon, and that CAP is going to bind. CAP is going to bind, similar to lac operon, just facilitate the operon's function. So if glucose then becomes present, you don't need that arabinose anymore. You only need 1 step to repress this operon, and that is going to be that same region. The ARA-C is going to bind its product, which is I, and that I is going to interact with oxygen, and it's going to prevent RNA polymerase from binding. So the exact same kind of conditions as the lac operon. It's going to use oxygen and its own product to prevent polymerase from binding, because now we have glucose again and we don't need another form of energy. Okay, so this is true or false. Does increased acetylation result in increased gene transcription? Good, that is true. Methylation decreases transcription, acetylation increases gene transcription. And that has to do with histone proteins, which is another concept worth looking at. Okay, and then we talk about HATs and HDACs. So HATs condense chromatin and HDACs decondense chromatin. Is that true or false? Good, false. It's reversed. HATs are going to decondense. HDACs are going to condense. And that has to do with acetylation and methylation as well. But these are just names for what they do. Okay, and what are cis-acting elements? Any idea? These are regions where trans-acting are going to be proteins. So this is kind of the small details within this chapter. So you can go into what the cis-acting elements are, but they are regulatory regions on the chromosome where trans-acting factors are proteins. And it's almost always true that a factor is almost always a protein. Another true-false. Promoters are upstream of the initiation site, while enhancers can be nearly anywhere on a chromosome. Good, true. You need the promoter to always be before where you want initiation to begin, because you want the enzyme to bind at the promoter and then get to the initiation site more downstream. But enhancers can be anywhere and influence almost anything. So as long as they're transcribed, they can get to where they need to be and change, increase, decrease transcription rates. Okay, and these are 3 DNA binding domains that he went over in class in his lecture. So HTH is called the helix turn helix motif, and that is going to bind DNA grooves. So those are the grooves in the DNA. Remember, you have major and minor grooves in DNA. Helix turn helix domains bind into those domains and either increase, decrease transcription. The zinc finger domain is going to use zinc atoms, kind of obvious. And it's going to bind to specific regions of DNA. So it can't just bind at a turn or a groove or any kind of area at once. It's specifically looking for a region of the DNA with the utilization of zinc atoms. And the leucine zipper is another one. And there's a protein that is going to promote dimerization. Dimerization is important because DNA polymerase 3 is a dimer protein. That's a protein that is used during DNA replication, and it actually comes in the form of 2 proteins interacting like this and moving along and replicating. So they're actually dimerization involves 2 proteins. That's why di means 2. And it's 2 proteins interacting with each other and moving along doing their function. And then the last part of this chapter talked about GAL. And the GAL information is an example of an operon-like system in yeast. So just some of the details like I covered in the 3 operons here is all you really need to know for the GAL information. Okay, any other specific requests for sections to go over? Replication, transcription, translation. Yeah. Yeah, definitely. This is all I have for biotechnology. Because again, it's kind of a 4-year information chapter. I mean, I don't think he's going to test you on the O.J. Simpson trial, more about the application of all of those things. So he might ask you to be able to determine a suspect if given a certain picture involving DNA fingerprinting. But the first thing he talks about are restriction endonucleases, which are used experimentally. And those are going to cut DNA at specific sequences depending on the specific endonuclease. So there's 2 types of ends they produce. There's going to be blunt ends and sticky ends. Which one is better? Good. Sticky ends are better because they're more specific. There's the overhang of nucleotides. that something else can come and anneal to, where blunt ends, it's just kind of making one stick to the other. There's not a whole lot of specificity there. So here there's kind of like an empty spot something has to bind to. Okay, so when we were talking about LAC-Z and XGAL and those experiments, at the end of that whole thing, do you select for blue or white colonies? Good. After all of that, all you're looking for is a white colony. And then what is the TI plasmid used for? Good. It's when you want to get genes into plants. So we've talked a lot about bacteria, but the TI plasmid is what you use to get genes into plants. It's how you create certain types of resistance in crops, stuff like that. They use TI plasmids to make crops resistant to pesticides and all that stuff. Okay, so DNA fingerprints are unique to every individual based on repetitive sequences called STRs, and those STRs are also known as what? Microsatellites. So just locations in the genome that are specific to every person, small little areas that are identifiable. And what is the AML test used for? Good. So you want to find out if your suspect is a man or a woman, run an AML test. And that's the end of biotechnology, because everything else goes into specific trials and stuff like that. But there's still 15 minutes left, so I can go back to yeah. Translation, yep, that's not too long, I can get through that. So translation is the process of going from RNA to protein. And starting off, true, false. A codon can code for more than one amino acid, and one amino acid is coded for by multiple codons. Okay, that is a false statement. The second half of that statement is true, but the codon cannot code for more than one amino acid. An example, something like GCU, that's not going to code for both histine and glycine. It's going to code for one amino acid every time without fail. But it is true that something like histine can be coded for by multiple codons. But that one codon is always going to code for that one amino acid. So that's why the genetic code is degenerate but not ambiguous. This is that reason. Okay, in a silent mutation, what does that result in? Anything go wrong in a silent mutation? Nope, everything works out fine. So you do have a new codon because something did mutate, but it's going to code for the same amino acid. So you kind of got lucky with that mutation. So if GCU codes for histidine and you mutate it to be GAU, but that also codes for histidine, yeah, you mutated, but it worked out fine, so you would never know that mutation is silent, everything is fine. What about a missense? What does that do? Good, it's the wrong or different amino acid. I think of it like you missed what you were supposed to be coding for. So it's a different amino acid. So you're supposed to code for histidine, you mutated, and now you're coding for lysine. Probably that's not going to go well. What about nonsense? Good. Nonsense begins with no, which means there's no more translation taking place. That means your codon turned into a stop. And lastly, a frame shift. What does that have to do with? Awesome. You either insert something new or you delete something. And instead of having your 3 reading frame, the codons, if you delete something like this, your reading frame shifts, and everything is read incorrectly from that point on. And what are your 3 basic requirements for translation? Good, you need a template, and that template is mRNA. What else do you need? How do you get the amino acids there? Good, you need tRNA. It's going to transport your building blocks. And then finally, you need something to do that all in. And what do you do it in? Good, a ribosome. It's important, you don't just need tRNAs, you need charged tRNAs. charged, meaning they have their specific amino acid already attached to them. Okay, and is it true or false that the anticodon loop of a tRNA base pairs with mRNA codons? True. That loop structure. Here is your anticodon region, and that should be interacting with your mRNA transcript. Which codon does the wobble hypothesis reference? Good, the third one. And here's just kind of a definition really quick, or an example. So if G base, we know G base pairs with C, but if it's in that third position, it could also bind with U, and it wouldn't really matter. It wouldn't care. So it's not a strict binding. So that third position is wobbly. It doesn't bind, I guess, strictly to the amino or the nucleotide it's supposed to. Okay, and here's another just information. Your tRNA must be charged, and it does so via amino acyl synthase with ATP, which means this process requires energy. And you have to add the correct amino acid to the 3' end, super important. And the, let's see, correct amino acid means there's only one amino acid per tRNA. And a tRNA doesn't just float around picking up whatever amino acid it wants. It's only specifically determined by this anticodon region. Always the same amino acid every time. The ribosomal sites are EPA, but the tRNA moves through them in the APE method. So remember that, just E always means exit, nothing's complicated with that, so E is always the last location. Okay, and the initiation steps of tRNA. Any ideas? What's the first step? You're working with mRNA at this point. And what does mRNA bind first? Does it bind to the tRNA, the large subunit, or the small subunit of the ribosome? Good. So it first binds the small subunit of the ribosome. And then what happens? Large subunit or tRNA? Good, tRNA, specifically what type of tRNA? What's it carrying? Good, you're carrying the formylmethionine. So formylmethionine is always the first tRNA to bind to mRNA no matter what. Methionine is an amino acid. Formula just makes it the first amino acid. And then lastly, what binds to start the process? Good, last part, the large subunit has to bind. True or false, the first amino acid, formylmethionine, that enters the ribosome begins at the P site? True. So in this case, this is kind of, it doesn't exactly enter the ribosome, and that's my fault for wording it kind of weird, because it sits down first before the ribosome comes in and kind of goes over it. So the first amino acid that actually does enter the complex does start at the P site, the A site, but the first amino acid that interacts with mRNA does so at the P site. Okay, I have 7 minutes, so any requests in the last 7 minutes? Yeah. Yeah, that's a good one. I was hoping to get to that. Okay, so I listed every enzyme I could think of that's involved in replication. And then it's important to know where they replicate, what they do, and what they do and where they do it, I guess. So DNA polymerase 1, where is that? It's going to work exclusively on the new strand of DNA. It shouldn't ever be interacting with the old strand. It shouldn't be anywhere besides where the new strand of DNA is being synthesized. And what's its main function? It has multiple, but its main one. Is it a polymerase or an exonuclease? It's an exonuclease. In what direction do exonucleases work in? 3 to 5, good. So it's a 3 to 5' exonuclease. That's its main function. What about polymerase 3? Where's that working? Good, again, it's at the new strand. It's actually making the new strand, which it has then 5 to 3' polymerase activity. Where's helicase? Is it in the middle of the bubble? Is it at the replication fork? Good, it's at the replication fork. So looking down here, it would be around here. And what's it doing? Good, it's unwinding, unzipping DNA to make it accessible for almost every other enzyme. And where is gyrase? Is it on this side of the fork or this side of the fork? This side, right? So it's ahead of the fork. And what's it doing? Yeah, it prevents supercoiling, so you can say, kind of like a made-up word, it's detensing the DNA. So it's making sure, I think of this like if you ever ride in a car with the top down and you have long hair, and you try to brush your hair out after you get out of the car and you start at the root, it hurts really bad because all your hair is super tangled. But if you start at the ends of your hair and work your way up, then that's what gyrase is doing. It's working at the ends and working its way back up, or helicase, I guess, is working its way back up to make sure there's no crazy breakage or tangling or anything like that. Okay, and ligase. Where does ligase work? Good, specifically it's on the lagging strand. And what does ligase do? Yep, it seals gaps. It anneals the DNA fragments. What are those fragments called? Okazaki fragments. Good. And primase. Where does primase work? Is it on both leading and lagging strands? Good. It produces primers for every piece, every Okazaki fragment, but it also starts the entire process of the leading strand. So there is one primer on the leading strand, so it is on both the leading and lagging strand. So you can say it's at the beginning of each fragment, as well as the leading strand, initially. And what does it do? Good, it puts down an RNA primer, because DNA polymerase III needs a primer to do anything. And then you have your single-stranded binding proteins, and where are those? Good, they're on the old strands, which means they are here already. And what do? Good, they stabilize them in order to keep the strands apart. Because DNA wants to be double-stranded, so without those single-stranded binding proteins, this bubble would collapse in on itself and reconnect. So these single-stranded binding proteins act as bumpers, so if this DNA tries to come back together, they're in the way and they can't re-anneal. I have 3 minutes left, so I'll take any questions. I also will keep this up here, so if you guys want to take pictures of anything. I also have, if you guys want to look at this entire review later on in the week or today, I have that URL as well. Oh, I'm sorry, did I skip stuff? Yeah, sure. Okay, so DNA polymerase has 3 to 5 exonuclease and 5 to 3 polymerase. Good, true. Both of them can do both, but they do have more specific functions. And then lastly, the PCR, polymerase chain reaction, what's it used for? Good, you're going to replicate DNA, and you're doing so with rapid DNA cloning. That's what it's used for when you want a piece of DNA to get to big levels quickly. Yeah. Oh yeah, sorry, I missed stuff. Okay, so is it true that heavier DNA moves towards the bottom of a centrifugation tube during centrifugation? Yep. You can imagine if you're on like a ride where it spins you super fast, the heavier you are, the more force you're going to feel pushing you back. And then what did Michelson and Stahl verify? Yep, good. The semi-conservative model of replication. Any other questions? What is it? You want me to do what? Okay, yeah. What does electrophoresis separate DNA on? Medium. Good, size. Bigger the DNA is, the harder time it's going to get, or going to have moving through that gel. Smaller DNA fragments can move through the gel very quickly. What does A bind to? T. G binds to C. How many bonds are there between A and T? 2, which means there are 3 between G and C, which means the more G and C bonds you have, the stronger your DNA is. Okay, I almost completed the page. So again, I will have this up here for a little bit. I will stick around for a little bit if you want to ask me any questions. But again, please use the QR code on all of the doors and rate today's session as well as your experience in Study Union. Thank you guys for coming. Oh yeah.